Hello again and welcome back to Science of Medicine. We talked about in the first part of pulmonary physiology about the anatomy of the lungs and also the process of ventilation and some of the influences related to that. This time we're going to talk about the gas exchange or respiration. Now, as you remember, we talked about this occurring at the alveoli. Oxygen is going to come into my body and carbon dioxide is going to go out. Those are necessary exchanges in order to have aerobic metabolism. Now at sea level, which is where I am, air is normally around 760 millimeters of mercury. Most of that is nitrogen, about 78%. 21% is oxygen. There's a small amount of carbon dioxide and other gases, excluding water. I live in a very humid climate, and so consequently water vapor will consume a bit of this partial pressure. This means that the partial pressure of oxygen in the atmosphere at sea level is about 160 millimeters of mercury. Now the partial pressure that reaches your alveolus is about 100 millimeters of mercury. Similarly, the partial pressure of carbon dioxide in the air is effectively zero, but inside the lungs is 40 millimeters of mercury. So I have a gradient, and that gradient will facilitate the gas exchange. On the alveolar side, I've got about 100 millimeters of mercury down in the alveoli, and on the venous side of blood, I've got about 40 millimeters of mercury for oxygen. So there'll be a spontaneous uptake of oxygen. And similarly, I have a gradient on the carbon dioxide with the bicarbonate leading to a carbon dioxide concentration of about 46, going into the lungs about 40. So remember on this idea of the alveolar exchange that this is highly vascular. So these capillary beds that are in these very thin-walled alveoli are going to facilitate fairly fast gas exchange. The exchange and uptake of oxygen is going to occur on hemoglobin. Now, when we were talking about the hematologic system, we talked about a hemoglobin being comprised of 4 porphyrin rings, these green structures here, each of which could take up a molecular oxygen. And in fact, they react cooperatively so that when one oxygen is taken up, it makes it easier for the second and then the third and then the fourth to bind. If I look at the details of this, if I look at this oxygen saturation curve here, the saturation going from 0 to 100% as a function of the partial pressure of oxygen, you can see that at 100 millimeters of mercury of oxygen, that the green hemoglobin A curve is effectively 100%, which means that it's saturated. There are 4 molecular oxygens on that molecule of hemoglobin. If you look at myoglobin, this curve in red, this is the transport or the oxygen containing moiety in the muscle cells. It also is saturated, but it's shifted to the left, meaning that except at this extreme of very high oxygen concentration or at the very low oxygen concentration, which is not reached in life, that oxygen will exchange spontaneously from the hemoglobin to the myoglobin, thereby facilitating transfer. I also show here a blue curve, which is for hemoglobin for fetal hemoglobin, and you can see that exchange from mom, hemoglobin A, to child, hemoglobin fetal, is spontaneous also. Now, I can modulate the amount of oxygen and its transport in hemoglobin by several things. And these modulations are very slight in the way in which they shift that oxygen saturation curve, but they're very important. So the few things that I want you to think about here that you can use to shift that curve have to do with the way that I remember it is in terms of exercise. So when I exercise, I actually want to basically be able to offload the oxygen to my blood vessel, to my muscles, so that they can actually carry out their metabolism and allow me to keep running or doing whatever I'm doing. that means that I need to shift my hemoglobin oxygen saturation curve to the right. So when I exercise, what happens? When I exercise, I normally heat up. So this red dashed curve compared to the blue one being when I'm resting, red when I'm exercising, or red when I've increased the temperature, show that I will actually then offload spontaneously the oxygen. Kind of a fallacy here, but if you think about it, just if it helps you remember that if you're exercising, you might have lactic acid produced from anaerobic metabolism, and so this will also shift the curve to the right. And the opposite of that is the green. So an increase in pH or a decrease in temperature makes it more difficult to offload the oxygen. The other thing that you should remember, and this is very important, is a small molecule called 2,3-DPG or 2,3-BPG. It goes by both names. It's a small molecule that when bound to hemoglobin will facilitate the curve shifting to the right. In other words, making it easier to offload oxygen. And in fact, this is one of the ways that we deal with changes in altitude. If I go from sea level up to a mile or 2 miles, I will need more oxygen in my tissues to be offloaded, and that's easily facilitated through this 2,3-DPG. That is a molecule that can be synthesized and upregulated very, very quickly and allow me so that I don't have altitude sickness. Now, I've mentioned to you before that carbon dioxide is transported in the blood by way of bicarbonate ion. And I wanted to show you how this actually is done. So we have carbonic anhydrase, which is present in the erythrocytes, in the red blood cells. And anytime you have carbonic anhydrase in the presence of water and carbon dioxide, it will facilitate the transformation into carbonic acid, the H2CO3. And this H2CO3 will then dissociate into bicarbonate plus a proton. It is what is determining the pH at physiologic pH, which is 7.40. This is considered to be sort of my Mason-Dixon line, if you will, for what is acidic versus basic in the body. I recognize that in aqueous solutions, that pH of 7.00 is what's defined as whether it's neutral, acidic, or basic. But from physiology, we consider that Mason-Dixon line, that set point of 7.40. And you can use the Henderson-Hasselbalch equation to relate these various components, the partial pressure of carbon dioxide, the bicarbonates, and the pH. Now, this is important because you can use acid-base chemistry understanding to make use of what's going on in the pulmonary and the respiratory system. And I've spoken about this in great detail in another video on interpretation of acid-base conditions. And I'm going to talk very briefly, or remind you very briefly, of some things as it relates to respiratory. So just to remind you that acids have a proton donor, which is there, and a base has a proton acceptor, like this lone pair on the nitrogen here. And as I mentioned to you, pH of 7 in aqueous solution is neutral, but in the body we will say that 7.40 is neutral, even though I understand as a chemist that that's not the case. Anything less than 7.40 is going to be considered an acidosis. Anything greater than 7.40 is going to be considered an alkalosis. So I have two regulators of my acid-base chemistry in my blood. The lungs, which are going to provide most of the change, regulation through carbon dioxide ventilation, which, remember, carbon dioxide, although it is not donating a proton, if there is carbon dioxide, water, and carbonic anhydrase, it will form the carbonic acid, and it'll do so on a molar basis, 1 mole of carbonic acid for every 1 mole of carbon dioxide. That means that I can either vent or retain carbon dioxide, and is doing so either have a respiratory acidosis or an alkalosis. We also talked in that interpretation of arterial blood gases about the renal changes, which are slower, which are regulated through creation of new bicarbonate, secretion of protons, and that's discussed in the other video on acid-base disorders. So, pictorial kind of thing, 7.40, is my set point. Anything with a pH less than that is acidosis. Anything greater is alkalosis. It's useful in terms of respiratory conditions to think about this in terms of a respiratory acidosis being caused by the retention of carbon dioxide. That leads to excess carbonic acid creation. And in alkalosis, the loss of carbon dioxide means I'm ventilating too much. There are several things that lead to this. In a respiratory acidosis, this can happen on an acute or on a rapid basis, such as if you have worsening of asthma or COPD, as well as neurologic disorders such as acute such as Guillain-Barré syndrome or amyotrophic lateral sclerosis or Lou Gehrig's disease. But there are other things. You could even have a stroke in your respiratory control centers, or even central nervous depressants like Ativan or alcohol. There are chronic conditions such as COPD or even poorly controlled asthma that can also lead to the buildup of carbon dioxide in your lungs over time. The kidneys will then make an adjustment for that, and that will lead to a chronic respiratory acidosis. Respiratory alkalosis is caused by loss of carbon dioxide in the lungs. That's from alveolar hyperventilation. And remember, it is impossible for you to breathe too quickly and too slowly at the same time, so there is no mixed respiratory acidosis alkalosis. To interpret these disorders, you need an arterial blood gas, which has pH bicarbonate PCO2. We will also use the PO2 when we're doing. Remember, we want this to be greater than 60. Normally, it's about 100 millimeters of mercury. You also will need to use an idea of compensatory changes. This is from Rose and Post's book, wonderful book, and but there are others that are on the internet that you can do, and this will allow you to determine if there are mixed disorders or it's a single disorder. I'll be back in just a little bit, and we'll talk more about problems as it relates to the pulmonary system. Again, you're welcome to like this or give me feedback, as well as subscribe to the channel as I post new videos. Thank you very much, and I'll see you soon.